Section 2 of Chapter 11 moves us into probability and Punnett squares. We are going to use probability to help us understand what the outcomes of genetic crosses will be, and we will use the Punnett square to help us visualize this and determine the offspring. Probability is the likelihood that an event will occur. So when we think of an example here, what are the odds of flipping a coin and landing on heads 4 times in a row? Well, if you flip a coin, you really only have 2 options. It's going to land on heads or tails. So the probability of the first flip is 1/2. If we multiply that by the probability of landing on heads in the second, that's also 1/2. So we're going to do this 4 times, and we end up with a 1 in 16 chance that flipping a coin will land on heads 4 times in a row. We're going to use the same idea with probability in our genetic crosses. So if we look at a Punnett square, this is the diagram that's going to help us use this probability to show what will happen when we cross different genetic variations. So it allows us to predict and compare. And that is the big piece for this. So Punnett squares will become your friends. We're going to use them quite a bit, but a couple of things about them. So our Punnett square, this is actually what the Punnett square will look like. On the top here is where you are going to have one parent, the genetic information, the alleles for one parent, and the other parent is going to be over here. So just a reminder, homozygous, a parent that's going to have a homozygous, they're going to have 2 of the same alleles. So think back to our pure breeding. These are the ones that had those same alleles. So in our pea plants, the capital T stood for tall. So a homozygous parent will either have 2 alleles for the tall or 2 of the short. So the capital T, the capital generally means dominant, and lowercase is recessive. So those are what we will put into the Punnett square. Heterozygous, on the other hand, they're going to have the 2 different alleles. So they will have an allele for the tall, for being tall, and 1 allele for short. 2 other things that we have to talk about before we actually get into filling out these Punnett squares. So the idea of phenotype and genotype. Okay, phenotype is the physical characteristic that is present. So this is that idea of the plants are tall or the plants are short. So it allows us to physically see how the gene is being expressed. So it's the physical characteristic. So phenotype, think of the physical characteristic. Genotype, on the other hand, is the genetic makeup, so the actual combination of the alleles. So in our homozygous, the genotype is going to have 2 capital T's. On the heterozygous, the genotype would be a capital T and a lowercase t. So the genotype allows us to see what the genetic makeup is. What is that combination of alleles? So phenotype, physical characteristics. Genotype, genetic makeup. So if we go on to our actual Punnett square. So this is the Punnett square, and again, the purpose of this is to help us to predict the offspring and then compare the offspring. So what we are going to do is we are crossing Mendel's F1 generation. So these were the hybrids from it, and they each ended up with capital T and lowercase t, which would be heterozygous. How do we start plugging these in? What you will do is you will put each of the parents on one up here and one down here. And you're just going to put in their alleles. So we have the capital T and lowercase t, capital T and lowercase t. So now what we're going to do is we're going to start to fill it in. So one way to look at this is we're basically just transferring this allele into each of the boxes. So we put the T here It's going to go here, and we're also going to then take this T and move it down. And generally we try and put the dominant first. You might have to make some changes after you put these in, but the whole basis of these is you're filling in, you're taking one from each. So this box would be one offspring, and it has one allele from this parent, and it has one allele from this parent. So if we continue down here, now we are going to take this allele and put it into each of the boxes, and we're going to finish up with this one. So our final Punnett square for this kind of set of offspring is going to look like this. This is our F1 generation when we crossed two hybrids, two heterozygous parents, and that's what we are going to get. What does this tell us about the genotypic ratio or the phenotypic ratio? So what do these look like? So again, the purpose of the Punnett square is to see these, to see what our offspring are going to look like. So what will happen is we are going to look at each of our kind of daughters, we'll call them offspring. So if we look at our very first square, we have a T and T. We have a homozygous. One of these. So that was this one right here. Notice now we have one that has a capital T and a lowercase t, and another one. So we would be able to fill this out as a capital T and a lowercase. So we have two of those. And in our final square down here, we have the homozygous. Two, it's going to want to auto-capitalize that for me. We are actually just going to have one of these. So that would be our genotypes. The genotypes for the offspring would be we'd have one that has the homozygous for the dominant, one that has the homozygous for the recessive, and one that's the hybrid that contains both of those. So what will our plants look like? So remember, these are based off of capital T was for tall, and then lowercase t was for short. So how many of them exhibit or have the tall allele? If we look in here, we have 3 that end up with the tall allele and only one that has both of the recessive short alleles. So we have a 3-to-1 ratio of our offspring when we crossed our hybrids. Let's continue with another example. So here we have a heterozygous and a homozygous with the recessive traits. Again, I'm just going to fill in my parents here. So put each of the alleles in a box. And now we are going to cross them. So I'm going to take this T and I'm going to fill it in both here. I'm going to take this T now and fill it in here. Now I'm going to take this T and fill it in here. And now this one comes down. And it doesn't really matter which order that you do it in, just making sure that the dominant allele is first in the box. All right, so what is our ratio, our genotypes for this? Well, if we look, we have 2 of the heterozygous that are going to have the dominant, one dominant and one recessive. And then in this one, we actually end up with 2 plants that have both have the recessive, so the homozygous of the recessive. So this tells us what the genetic makeup of these plants are going to be. So what are these plants going to look like? What are their phenotypes? How many tall are we going to have? And how many short are we going to have? Our tall, and the tall, anytime the dominant gene is expressed, that's when we will get that. So we only have 2 that have that dominant gene. We also have 2 that have both of the recessive genes. So this is going to be a 1-to-1 or 2-to-2 ratio for the offspring of this cross. Okay, one more example here. Now we're going to use the actual terminology. So you should be comfortable with using the alleles and then also kind of using the vocabulary to understand what the alleles will be. So a homozygous dominant. What does that mean? A Homozygous dominant means that we're going to have two of the same alleles, and they tell us that it's the dominant. So I could put the capital T's in here. They also tell us that it's going to be crossed with a heterozygous. So hetero is going to have one of each. So we'd have a capital T and a lowercase t. And now I'm just going to go ahead and fill these in. Again, it doesn't matter which direction as long as you take one from each. Now I'm going to go this direction. And we have T and the T. So this is what your Punnett square should look like when you crossed a homozygous dominant and a heterozygous. So what would your genotypic ratio be? Well, we are going to have 2 of the homozygous dominant, and we're going to have 2 of the heterozygous. What will things look like? So remember, when genes are expressed, if the dominance is there, that is what we're going to see. So we are going to have 4 tall. And this is how we use Punnett squares to help us determine what will our offspring look like, phenotype, and what is their genetic makeup.